champions, I hope that you are doing great and I welcome you all to mock test 6 of our NEET Premier League series. And today, children, as I already told, 20 questions. The majority of the questions will be from the chapter that we are already doing now, that is molecular basis of inheritance, and rest 10 questions will be from the other chapters that we have already completed, like human reproduction, reproductive health, and what? Human health and diseases, principles of inheritance and variation, sexual reproduction in flowering plants. So little questions will be there. So are you ready? Let's start. And please do not watch the video without giving an attempt first. So please attempt it first, and if you're not able to understand or if you want to check your scores, then here I am. You can tally your answers with me. Chalo. First question, which of the following statement is correct? Adenine pairs with thymine through 2 H bonds. Okay, so since it is about nucleotides, I want to say that there are two types of nitrogen bases. One is purine, one type is purine, another type is pyrimidine. Cool? Now, under purines comes adenine and guanine. Under pyrimidines comes thymine and cytosine. So basically, adenine is the one that pairs up with thymine and guanine is the one that pairs up with cytosine. You can remember that the GC bond, since they are looking kind of very similar, the bond is also very close. Okay, they are very similar. We say, the people who are close, they kind of start looking like each other. So yes, just like that, they have 3 bonds. And what are these bonds, children? Both of these things that you are seeing over here, this bond, everything is what? Hydrogen bonds. So let's now see the answer. Adenine pairs with thymine through 2 H bonds. Which one is correct? Adenine pairs with thymine with 2 H bonds. It's already correct, but still. Adenine pairs with thymine through 1 H bonds. Wrong. Adenine pairs with thymine through 3 H bonds. Wrong. Adenine does not pair with thymine. Absolutely wrong. So this is the answer. This came in NEET 2020. Such an easy question for marks, very easily. First question done. Second question. Purines found both in DNA and RNA are. You are asked about purines which are found both in DNA and RNA. Again, I'll tell you one more thing over here itself. So, purines are the same, okay. I'll write same for DNA and RNA, okay. A and G will be there in DNA as well as RNA. But when you talk about pyrimidines, not same. For DNA and RNA. So, in the case of RNA, okay, in the case of RNA, this D is replaced by U, that is uracil. So this is thymidine, this is thymine, sorry, and this is what? Uracil. In RNA, you have uracil. So in total, how many pyrimidines do we have? U, T and C. Whereas in the case of purines, how many of them we have? 2, A and G. And it is same for DNA and RNA. The question is about purines. Purines found both in DNA and RNA are, children, cytosine and thymine. Is it so? No. Adenine and thymine? No. Adenine and guanine? Yes. A and G. A, G, O, G. You might have listened that song. I don't want to sing here. So it is NEET 2019 question, very much important question. Okay. Again, very easy question, not important. I'll say easy. Going ahead, which one of the following does not follow the central dogma of molecular biology? What does central dogma say, children? Central dogma says the DNA is basically, DNA has to make proteins. Okay. I'll make it easy for you. DNA has to make proteins, but DNA cannot go outside the nucleus for making proteins. And the ones that make proteins are outside the nucleus. So the problem is, DNA is inside the nucleus, but the one that makes protein are outside the nucleus. That is our ribosomes. So the DNA, can it go outside? No. What can it do? It can get converted, not converted. Basically, it is, I always say converted, I don't know why. So DNA's information is used to make something that is known as RNA, especially messenger RNA, and this RNA helps in the formation of proteins. This process that you see over here, it is known as transcription, and the messenger RNA's message is understood with the help of translation. It speaks in different language, proteins understands in different language, so basically it has to translate. So translation happens over here, okay. And one more thing, the DNA, when we want to make more copies of DNA, what do? We do something, we make more DNA with the help of something that is known as replication. So this was given by our own guy, that is Francis Crick, who was the person who found the double helix structure of DNA along with James Watson. So Francis Crick was the person who gave the central dogma. And now what's the question? Which one of the following does not follow the central dogma? P, Mucor, Chlamydomonas, HIV. It's HIV. Why HIV? Because HIV is a virus, human immunodeficiency virus. And you know what does it do? You will be very much, you already know it, you are my children, right? We studied it in human health and diseases. So, they have an enzyme which is known as reverse transcriptase. Reverse transcriptase. What does it do? Reverse transcriptase reverses the whole process. It makes DNA out of the RNA. Because HIV, we have DNA, right? And if it wants to infect us, it has to be like us. So, the RNA that it has, it converts into, it helps in the formation of DNA, and this helps, this is known as reverse transcription, and the enzyme that helps is known as reverse transcriptase. So, according to central dogma, things has to flow like this, but over here, this is ulta that is happening. So, which is why HIV is an exception. Works? Question number 3 done. Kevin NEET 2019. Going ahead, the 3' to 5' phosphodiester linkage inside a polynucleotide chain serves to join. What do you mean by 3' to 5' linkage? Listen to me carefully. So, we have a pentose sugar over here. In the case of DNA, RNA, everyone, okay. Pentose sugar, why? 1, 2, 3, 4, 5 carbons are there over here, children. Okay, we have 5 carbon, one is outside only. Okay, in the case of RNA, they have an OH group over here. DNA does not have OH group over here. Okay. Now, on the 5th carbon, so I am writing carbon also here, okay, so that you understand. On the 5th carbon, we have a phosphate group attached. Okay, we have phosphate group being attached. So, it is known as 5' end. If this end is free, you call it 5' end. Okay, but fortunately, what happens is that, oh, I actually draw it a wrong place. So, just imagine this is over here, okay, and we have, oh, ma'am being ALSI does not work, so ma'am has to make it again. So, I will make it again for my little champions. 1, 2, 3, 4, 5, and over here I have the carbon which has the phosphate group. Okay, on the 5th, this is 5th. So, it is on a 5' end if it is free. But what happens is that there will be another pentose sugar over here. Same thing happens over here. It will also have phosphate group attached. But this phosphate, no, it is not free. It attaches to the 3rd carbon over here. So, this is 3 and this is 5. This is 3', this is 5'. Over here the 3' has OH group through which it is attached. So, basically this C also has an OH group onto which the phosphate is attached. Similarly, here also an OH group is there onto which the phosphate is attached, which is why these bonds, this is one ester bond, this is second ester bond, together you call it diester bond. Diester bond, but since phosphate is also involved, I call it phosphodiester bond. I call it what? Phosphodiester bond. So, you tell me, this is one nucleotide in total, okay. Nucleotide has a nitrogen base I am making over here. Nitrogen base is on the 1st carbon, okay. So, this is a whole nucleotide. This nucleotide and another nucleotide are attached with the help of 3', 5' phosphodiester linkage, okay. So, over here you see, what does it do? Basically, it attaches one nucleotide with another nucleoside. Is it like that? Does it attach one nucleotide with another nucleoside? Don't know. It's basically nucleotide. That might have been a printing error. This is one nucleotide to another nucleotide. Works? Okay. So, this is the correct answer. Going ahead. Antiparallel strands of DNA molecule means that one strand turns clockwise. Okay, antiparallel, we say that DNA runs antiparallelly. That is, I told you, if the 5' end is free, it will be over here. Okay, if the 3' end is free, it will be over here. Similarly, 5' end is over here. 5' end is over here, 3' end is over here. This is antiparallel. 5 to 3, 5 to 3. Ulta is happening over here, right? What do you mean by 5' end? I told you already, when this is one strand, children, which has the nitrogen base, and this is the carbon with the phosphate. Okay, this is the 5th carbon. So, the 5' end, if there is no one to attach upper, it will be free. On the other hand, this also has the opposite one, where the sugar is ulta. Ayo. Yeah. Okay, this is the 1st carbon, 2nd, 3rd, 4th, and this is 5th. So, over here if you see, the 3rd carbon if you tilt it over here, you'll understand the third and this attaches. But over here, the third is free. And third has what, children? I already told you it has OH group. I twisted it, so OH will be upper, right? So similarly, OH here will be on the upper side, okay? And here I have NB on the first. Okay, now you only tell me this is 3' end and this is 5' end. This is 5' end, this is 3' end. Same thing happens. Similarly, the whole strand goes like this only. So if this is straight, this is ulta. This is why we call it anti-parallel. So 5 is to 3, one goes, other one goes from lower 5 is to upper 3'. Okay. So read, one strand turns clockwise. Is it because of that we say it is anti-parallel? No, children. One strand turns anti-clockwise? No. The phosphate groups of the 2 DNA strand, that is over here. So 5 is basically for P and this is for OH. Okay, the phosphate groups of 2 DNA strand at their ends share the same position. Are they sharing the same position? No, I see phosphate over here and OH over here. This is wrong. The phosphate groups at the start of the 2 DNA strands are in opposite position. Yes, they are in opposite position. So this is the correct answer. Came in 2006. Cool. Going with the next one. The following ratio is generally constant for a species. A plus G is equals to, okay, they are saying constant. So basically it has to be 1. So can A, I'll tell you one very important thing, okay, before starting this. There is a rule, that is Chargaff rule. What is it? Chargaff rule. According to it, it says, okay, however amount of A is there, that's the same amount of T. However amount of G is there, that's the same amount of C. Okay, so for example, 20% of A is there, that means 20% of T will also be there because they are complementary to each other. So when A is there, A will obviously bind with T, so that will be the same amount. Similarly, if this is 20%, next will be 30%. Why did I say 30%? Because you have to understand the fact that 50% of the DNA has to be purines, other 50% has to be pyrimidines. Okay, other 50% will be pyrimidines. So obviously, if A is there, this is a purine, the other one is pyrimidine. So 50% if it is this, 50% will be that. That's how it becomes right. Fine. Now, if it is 70, then it will be 33. You have to calculate, you have to make it 50. That's all. But people should not do like this. That is, this is 20 and that will be 30. No, children. A, that whatever amount of A is there, that much amount of T will also be there. Now we want constant. Now we want things to be constant. For that, what we will do? If you suppose, if I write A plus T is equals to G plus C is equals to 1, will that be fine? No, it would not be fine. How can this be equal to 1? So over here I am saying 20, 20, so 40. Over here G and C is basically 30, 30, so basically 60. Is it equal to 1? No, but on the other hand, if I take like this, okay, if I take like this where A plus G divided by T plus C is equals to 1. Is it correct now? Okay, this is 20, this is 30, this is 20, this is 30, 50 by 50 is equals to 1. So, the amount of purines by pyrimidines will be a constant. The amount of purines by the amount of pyrimidines will be constant. Please remember that this is Chargaff rule, second rule, okay? Purine, pyrimidine is equal to 1. Okay, so now let's check for it. The answer is this. You want a constant? The answer is this. Okay, question number 6. Then 2004 question it is. In a DNA, percentage of thymine is 20, then what will be the percentage of guanine? You tell me. If I am saying the percentage of thymine is 20, okay, what will be the percentage of guanine? So if thymine is 20, then thymines those, who is thymines those? Adenine. Adenine will also be 20, right? Now, if obviously if thymine is 20, okay, rest total we have to make 50%. Cytosine will also be same like guanine. So if you find the cytosine's amount, then only you can find. So if thymine is 20, cytosine will be what? 30, children. So this guanine will also be 30. Okay, you got to know everyone's. Cytosine will be, okay, cytosine will also be 30. This is a purine and this is a pyrimid. Oh, for your, okay. Listen, okay, I don't want to confuse you guys. This is thymine, 20% given, okay. If thymine is 20%, then Cytosine, which is again another pyrimidine, it has to be 30 because pyrimidine should be 50, purine should be 50. If this is 30, then its partner, that is G, will also be 30. So the answer will be what? 30%. Okay. If you are asked about adenine, then this is 20, then adenine will also be 20. Got it? Now going ahead. Length of one loop of DNA is, what do you mean by one loop? So if suppose your DNA is like this, this thing is known as one loop, children. This whole thing is known as one loop, and I am saying basically it has 10 base pairs. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So each loop basically has what? 10 base pairs. What do you mean by base pairs? A, T, G, C. That is what I mean by base pairs. And in between each base pair, the space which is there, it is 0.34 nm. And if 10 base pairs are there, 10 into this is 3.4 nm. We can directly remember this, that is also fine. Now if they are asking in Armstrong, it will be 34 Armstrong. This is another unit. So total length of loop is 3.4 nm. Please remember length of one loop or pitch, if they are asking the same thing. Okay, it will be 3.4 nm. Okay, this is 34 Armstrong. I have written like 84, so I will just make it correct. Cool, my champions. 9th. An octamer of histone complex with DNA forms. So this is an octamer of histone. Okay, octamer as in you need how many? 8. So 1, 2, 3, 4. I mess with the 3D things, so please forgive me, but yeah. Suppose this is 1, 2, 3, 4, 5, 6, 7, and in lower area there is 8. Okay, 8 histone, and your question is are complex with DNA. So the DNA turns around, okay, and almost 200 base pairs are there over here itself. Please remember that also. In this turn, you find 200 base pairs of DNA in all the nucleosomes. Okay, so this structure that you see is known as nucleosome. I already answered it, but yes, give me 90-90. It's a very easy question, right? Okay, going ahead. In DNA, when AGCT occurs, their association is as per which of the following pair? So AT and GC. That is the thing. So it's given over here. The same thing. Okay. Now, in prokaryotes, the genetic material is, okay, tell me, in the prokaryotes are the one that does not have nucleus or do not have membrane bound stuff. Okay, in their case, for example, bacteria are coming under prokaryotes. They are the only one that comes under prokaryotes. Okay, in them they have, in us we have linear DNA. Okay, but in the case of bacteria, they have circular DNA. They have circular DNA, and the circular DNA does not have histone, children. No histones. But we have linear DNA, and we also have histone proteins because we want to make it everything compact. Okay, so this is about humans, eukaryotes. I'll just write humans as of now. Cool. So circular DNA without histones. Works. Going ahead. Genes are packaged into a bacterial chromosome by. Genes are packaged into a bacterial chromosome by. Obviously, how can it be packed? What do we need? Histones, so it's not there. Other proteins can be there. And children, genes as in the DNA. You're asking about DNA. DNA, it is acidic in nature, children. Okay. So if you want to pack acidic things, you need basic proteins. You need basic proteins. Even histone is a basic protein. Okay. So please remember, this is the correct answer. 1997 question. Easy it is, no? Having fun? Okay. The experimental proof of semi-conservative replication of DNA was first shown in, okay, the semi-conservative replication experiment was first shown in what? So this was done by Michelson and Stahl, Michelson and Stahl, and it was on none other than, what do we have over here? We have bacterium, that is E. coli. Okay, this is the answer. Came in 2018, very easy question. Can you imagine these type of questions coming for your exam? You can easily score marks. There is no point of losing marks at all. Easy, direct question, no? Now, the semi-conservative replication of DNA was first demonstrated in, now they have asked the name, I told here bacterium, what bacterium? E. coli. So, Escherichia coli, 2009 question. See how it is repeated in different format. Okay. Now, the unequivocal proof of DNA as genetic material came from the studies in, I repeated this question because I wanted you to remember. So, everyone said that the DNA is genetic material. Okay, the first person to come was Griffith. Okay, with his experiment, then came Avery, MacLeod and people like that. Then came what? Our Hershey and Chase. And they finally, okay, they finally said that yes, this is how we can prove that DNA is a genetic material, and it was done on with the help of a bacterial virus. The name was bacteriophage. The name was bacteriophage. Okay, this is a bacterial virus. Why do I call it bacterial virus? Because this virus goes and infects the bacteria. Goes and infects bacteria. This is a virus, children. Please remember. Okay, so it is a bacterial virus that is given over here. This is the 15th question. Now, next question. The transforming principle of pneumococcus was found out by Avery. As found out by Avery, MacLeod and McCarthy was what? So, according to McCarthy and Avery and MacLeod, the transforming principle, so this was done by Griffith. Griffith did a pneumococcus experiment where he used 2 strains of the bacteria pneumococcus and he infected the mice. He found that S1 was the one that causes pneumonia, whereas R1 was non-virulent. But then he did one thing, he heat killed S and then he put on the mice, it didn't work. Okay, the mice didn't die of pneumonia, it was live. But when he put heat killed S and live R in the mice, he found that the mice died because the live R which was there, it is getting transformed into something else, that is S variety. This was heat killed, but it was passing down something to the live R which made it different. And the experiments were done by Avery and MacLeod. They found that they kept on doing this. Okay, what they did was, for example, this is a heat killed S1 and this is an R1. Okay, they treated this with protease. When protease was treated with protease, still it was getting converted to S. Okay, that is the S strain. When it was treated with RNase, still it was getting converted to S. That means RNA and protein, even after getting damaged, still somehow it is getting converted into S. But now when they were treated with DNase, this was treated with DNase, the DNA got damaged and hence this particular transformation was not seen. The R strain remained to be R and the mice was remained to be living. So it was them who said that DNA is basically the one, but still people didn't believe it, which is why they went ahead with Hershey and Chase experiment. Okay, going ahead, the pneumococcus experiment proves that, just now I told, the pneumococcus experiment, basically the part that was did by Griffith also, it was initially done by Griffith and then this was done by Avery and MacLeod and everyone. So all of them, they were saying that DNA is a genetic material, DNA is a genetic material, that was the only thing. So over here I won't even read the option, this is the answer, children. Okay, the bacteria do not reproduce sexually, no. RNA sometimes controls the production of DNA and proteins, no. DNA is genetic material, yes, that was said by them. Okay, bacteria undergo binary fission, no. C is the correct answer. Got it? Going ahead. Escherichia coli, fully labeled with 14N medium. The 2 strands of DNA molecular of the first generation bacteria have. Okay, wait. Okay. Escherichia coli fully labeled with N14 meat. Okay. So basically, it was like this. Escherichia coli fully labeled with N15 is put in N14 medium. The two strands, okay, of the DNA molecules of the first generation bacteria has what? So basically, just imagine that you have the N15 one, okay, the normal one, but now you are putting this Escherichia coli in a medium that is having N14. Okay, so obviously when they replicate, it will open up. So this opens up into two strand, and correspondingly there is semi-conservative model experiment, right? So the next strand that will be formed will be having N14 because DNA has nitrogen in them, okay? So they will use this nitrogen for formation of other DNA. So one strand will be 15, other strand will be 14, this strand will be 15, other strand will be 14. So don't you think that this is hybrid? Yes. So according to the experiment done by them, it was like the one that had 15 and 15 like this, okay, they will be in the bottom because it is high density isotope of nitrogen. Now one that has 115 and another 14, it will be in the middle, intermediate level, and one that has light, that is both of them are 14, this will be at the top. Over here you see the different density is there, and also it is does not resembling its parent. The parent has both green, over here it's not like that. So they have different density and do not resemble the parent DNA. 1992. Okay, so please remember this is the answer. Going ahead, nucleotide arrangement in DNA can be seen by. So you remember Watson and Crick, James Watson and Crick, okay, they worked on the Morris Wilkins and everyone, Morris Wilkins, right, and Franklin, Morris Wilkins, yeah. So basically all of them, they gave the X-ray diffraction thing, data, right? So X-ray crystallography was used for finding this thing, okay, and based on which James Watson and Francis Crick were finally able to put down the DNA double helix model, okay. Now going ahead, DNA is mainly found in nucleolus, nucleus, cytoplasm, none of this. What do you mean by nucleolus? So basically this is the cytoplasm, this is the whole cell, this area is the cytoplasm, okay. Inside this, what do we have? This is the nucleus. Inside the nucleus there is a dark spotted region which is known as nucleolus. Your DNA is in the nucleus, okay. Mainly the DNA is in the nucleus. If you are asked about eukaryotes have DNA in the mitochondria also, but that's not an option over here. That is known as maternal DNA, because we assume that mitochondria and everything existed autonomously, they were alone, but then once they came inside the cell, they started living with us. So mitochondria has its own set of DNA, our nucleus also has its own set of DNA. Here the options are not like that, so we'll go with just the nucleus, okay. 20th question. Now it's not from these chapters, now it is different from different chapters, okay. So an abnormal human male phenotype involving an extra X chromosome is a case of. So usually what happens, you have X and X chromosome from the mother, X and Y chromosome, in that one can come to a gamete, right. So in one gamete usually one comes, but over here there's a change, and the change is that both the chromosomes, they do not fail to segregate, so basically they come to get together, and in another gametes that are formed there is nothing. Okay, here also there is X chromosome and there is Y chromosome. Now assume that this and this fuses. You get XXX, that is super female. When you cross this with this, it will be XXY. So XY is usually male. When Y is there, that means it has to be male, but there is one more X. There is one more X. So instead of having 46 number of chromosomes, you have 47 number of chromosomes there. One extra is there, okay. And this is known as Klinefelter syndrome. This is known as, children, Klinefelter syndrome, where it is a male body but with certain characteristics that matches the female, okay. So Klinefelter syndrome is the answer. Going ahead, a person whose father is colorblind marries a lady whose mother is daughter of a colorblind man. What? Let's see. So a person whose father is colorblind. Okay, father is colorblind. That means here it has Y. marries a lady now listen to me very carefully, okay? It's talking about the son of this father. Can the son ever receive the XC over here? No, because the son is formed with the help of XY. The son is formed with the help of X and Y, right? So here the father, basically for giving the son, they have to give the Y chromosome only. The only thing, if it's like this, XX and XY, I have to make a son, okay? So if I cross this and this, I get XX, this is a daughter, okay? If I cross this with this, this is a son. Now if I cross this with this, this is a daughter, this with this is a son. But nowhere X is passed down to the own son. No, X is not passed down to the own son, it is to the female that is passed down. So the son is safe, son is not color blind. Not color blind. And now it is said that the son, basically a normal son, is marrying a lady whose mother is daughter of a color blind man. Mother whose, okay wait, marries a lady X, okay, whose mother is a daughter of a color blind man. Okay, whose mother is a daughter of the colorblind man. So basically she is a carrier. Colorblind man always gives excess. So this is there. So here also, here she can be a carrier. Here she can be a carrier. This particular lady can be a carrier. Now when they fuse, okay, normal X, XY and XC, when they fuse, okay, what will be happening? All sons colorblind, all sons normal, and some colorblind, some sons normal, some all colorblind, all daughters normal. So let's see. So over here, if I cross this with this, I get XX. If I cross this with this, I get XCX. Okay. If I cross this with this, I get XY. If I cross this with this, I get XCY. Okay, I see that one son is having this, one son is not having this. Okay, now you're asked about the children who are colorblind. So, all sons are colorblind, no children, all sons are not colorblind. Or some sons are normal and some sons are colorblind, yes. Some is colorblind, some is normal. Now, all colorblind, no, all are not colorblind. All daughters are normal, no, all daughters are also not normal. So, basically this is the answer. How did I find it, children? Listen to me. They said about the father first. A person whose father is colorblind. So, if father is colorblind, it cannot be passed to the son. It can be passed to daughters, it cannot be passed to son. So, son is normal. Now, over here, there is X, whoever is there, A, I am putting A, okay. It is written, it marries a lady whose mother is a daughter of a colorblind female, a colorblind man. If colorblind man is there, then it is passing at least one X chromosome to the daughter. So, this daughter, whoever is there, will be a carrier of color blindness, okay, because another X is there to dominate this, so it won't be showing up. That person will be a carrier. Now over here I am making a daughter again. Anyways, whatever you do, there are chances that either they will be normal, but the daughter can be like this also. I will take this condition. I will take this condition. I will not take the normal one, because here I am talking about color blindness. So I will take the abnormal one, and then I will do the crossing, and I will find the maximum possibility that could come is this. Okay, when there is XCY, there is no one to dominate, so this has to be colorblind. When it's normal, it's again a carrier because X is there, normal X is there, so it will cover up. Here also, it will be normal. So basically, this is the answer. It came in 1996. It's a mind-ticking question, yeah. The polygenic genes show. What do you mean by polygenic? Polygenic, basically, polygyny means there will be different set of genes for which will be determining one character. There will be different set of genes which will be determining one character, children. Okay, so A is different karyotypes, different genotypes, different phenotypes, none of this. So characters means something that is known as phenotypes, not as genotypes. For example, for skin color, you have different genes which will be giving the skin color. Skin color is basically a phenotype. So this is the Answer for the same. Cool? Works? Going ahead. In which one of the following both autogamy and geitonogamy are prevented? So, all of them, they are monoecious, but you're talking about papaya, it's dioecious. What do you mean by dioecious? Here, there will be one sex in one plant. So, for example, this is female. The whole flowers which will be over here will be females only. And what do you mean by autogamy? Autogamy is when self-pollination. Basically, when you have flower, and this flower basically has the male, the female and the male thing, so that it can go, this is known as autogamy. But when in the same plant from one to another pollination is happening, it's known as geitonogamy. But you see both are females, what will you pass on? So it is papaya, which is dioecious. Okay, works? Going ahead, which one of the following pairs of plant structures has haploid number of chromosomes? You have to find haploid number of chromosome. Haploid number of chromosomes are only found in the embryo sac. Okay, an embryo sac has these many cells. Simple. Okay, if you talk about embryo sac, it has these many cells. One is the egg cell. Okay, the other ones are the synergids. So these are the synergids. This is the egg cell, and then they have antipodal cells and two polar nuclei. Now match what one is there. They are all haploid. So nucellus and antipodal cell, no, nucellus is not there. Egg nucleus and secondary nucleus, no. Megaspore mother cell and antipodal, oh my god, no. Egg cell and antipodal cell, egg cell and antipodal cell matches the thing. Tick. Easy. Okay, do not get confused by reading big things then. You know what is haploid, you can make it. So this is the embryo sac, and embryo sac has everything that is haploid. Works for you? Going ahead. 26th, you have to find the correct one. Okay, so A is given. A is supposed to be primary follicle, but over here I do not see anything to be like that. Primary oocyte and everything is given, but it's not there. Okay, so what about them? Over here B is given, and B is given to be corpus luteum, and B is corpus luteum is correct, because you will here see the secondary oocyte has come out, that is ovulation has taken place. Now what is left behind, children? The left behind thing is known as corpus luteum, and corpus luteum is the one that secretes progesterone, which maintains the endometrium lining. So this is the correct answer. Always remember the picture that is after ovulation, that means it is corpus luteum, and corpus luteum is the one that secretes progesterone, which maintains the endometrium lining as well. Okay, fine. Going ahead. In if mammalian ovum fails to get fertilized, which one of the following is unlikely? If ovum is not fertilized, which one of them is unlikely? I'll tell you when it is not fertilized what all happens. Okay, so when it is not fertilized, initially we have corpus luteum, which is formed after the ovum is released, right? So corpus luteum secretes something that is known as progesterone, and it also secretes something that is known as estrogen, both of them that maintains the endometrium. But when progesterone is being made, it goes and gives an inhibitory effect to GnRH, that is gonadotropin releasing hormone. Now GnRH says, hey, LH and FSH, stop your secretion. Now when LH stops, it says to corpus luteum, you also stop. Now if corpus luteum stops, progesterone will also be stopped. If progesterone is not there, endometrium lining will not be maintained. And similarly, estrogen also stops. Okay, hence endometrium lining is not maintained and the flow happens. So that means ovulation, fertilization has not taken place. But if fertilization has taken place, to maintain the corpus luteum, okay, there is another hormone that comes in, that is HCG, human chorionic gonadotropin, that comes to play. So instead of LH saying you to maintain, you to get maintained, that is corpus luteum to get maintained, HCG says, hey, you have to maintain yourself, you have to keep producing progesterone and estrogen for the endometrium formation if pregnancy has taken place. But since pregnancy has not taken place, this is not happening. All this is happening. Progesterone going and saying that GnRH, please stop your secretion. GnRH is telling LH and FSH to stop your secretion. When LH stops, corpus luteum degrades. When corpus luteum degrades, progesterone also goes down, estrogen also goes down, endometrium flows down. So corpus luteum will disintegrate, it will happen, children. Progesterone secretion rapidly declines, it will also happen. Estrogen secretion further increases, how can that happen? It also decreases, right? So this is wrong, it is unlikely. Primary follicle starts developing, that is also there. When endometrium flows down, another follicle starts developing for the next round, okay? Then, going ahead. Capacitation occurs in, what do you mean by capacitation? So capacitation is where, so sperm, no? You know sperm has middle piece, head region and the whole tail and everything, right? So middle piece has the nucleus and at the top it has the acrosomal cap, right? So, and the whole body is basically covered by something that is known as plasma membrane. The whole body is basically covered by plasma membrane. So now, if, just think that you have to release the enzyme that is there on the acrosome so that it can penetrate inside the egg. Otherwise, it won't be able to penetrate inside the egg. So what do we do? We make certain changes in the plasma membrane such that this acrosome can easily release its enzyme, and this is known as capacitation. This changes in the plasma membrane is known as capacitation, and it occurs in the female reproductive tract. Okay, so the answer is this. Works? Next question. Select the hormone releasing intrauterine devices. What do you mean by hormone releasing intrauterine devices? So they bring out changes in the uterus such that implantation does not take place. Even if fertilization has taken place, implantation does not take place. So the uterus becomes hostile. In a way, it won't allow anything, the blastocyst to come over here and sit over here and basically develop itself. There are two types of hormones over here. It is given progestasert, that is one hormone releasing intrauterine device, then is LNG20. You have three different types. One is physical one, which is lipase loop. Okay, other one is copper releasing one. Okay, which is multi-load 375, then copper T, all these things. Okay, they release copper, and copper basically makes it very difficult for the sperms to move in. Okay, and also it brings out changes in the endometrial lining such that implantation does not take place. Progestasert and LNG20 also work on the same principle. They do not allow the implantation to take place there. Okay, it makes a lot of changes. So these are the hormone releasing ones, IUDs. Fine. There's only one, and I don't know, the typing person has done so many mistakes. Okay, which of the following is incorrect regarding vasectomy? Vasectomy means what, children? Remember, we have the testis. The testis has seminiferous tubules, testicular lobules and everything. So there are testicular lobules. These testicular lobules have seminiferous tubules which open into something that is known as rete testis. Rete testis open into something that is known as vasa efferentia. Vasa efferentia opens into something that is known as epididymis. And epididymis basically opens into something that is known as vas deferens. In the case of vasectomy, this vas deferens is cut and it is tied. Okay, so no sperm occurs in the seminal fluid. Is this the case? You are asked about incorrect thing. Seminal fluid is basically when the secretion. Okay, if you go ahead, you will be finding there will be so many glands which will be releasing certain things for receiving the sperm, and this all things together you call it as seminal plasma. Okay, seminal plasma plus sperm is equals to semen. Now it's saying no sperm occurs in the seminal fluid. Yes, no sperm occurs. Fine, it's not there because it has not been taken. No sperm occurs in the epididymis. Can that be true? If I cut from here, sperm can still be occurring over here, right? Epididymis will still have the sperm, so it's fine. Vasa differentia is cut and tied. You're asking about incorrect. Vas deferens or vasa differentia is the same thing. It's cut and tied. Irreversible sterility. It is incorrect or not? No, it's correct. It is irreversible sterility. So no sperm occurs in epididymis. That is wrong, children. Okay. So please have a note on that. And with this, we finish the 6th mock test. I hope that this particular video was just a second. Okay. This particular video was really helpful for you. And if you think that we are helping you really well, then do not forget to subscribe to our channel. And also, if you have doubts regarding any question that I have discussed over here, you can feel very free to put it down in the comment box. I will be clearing each of your doubts. Okay, you're my own children. I will be doing that for sure. Now that's all, children. I hope you had a happy Dashera as well, and I wish that you always remain blessed. Bye bye. Take care.